So today I'm really excited to tell all of you about some ongoing research and recent work in our group. And this is basically done in collaboration with my good friends, Ray Robertson Anderson, Michael Rust, and Jenny Ross. In fact, this collaboration started. So every science story has also a really often also has a nice meet cute story. So our story is that we all met up in the biosphere in Arizona about 5-6 years ago. And this was a conference where basically the goal was that you discuss science with your colleagues and pitch sort of pie in the sky ideas of what kind of science you would like to do, right? And the idea that our team came up with was making materials, right? So synthetic smart materials that can change their state and change their properties on a time schedule. And we were going to do that by combining sort of biopolymer materials with sort of with clock proteins from bacteria. So I'm going to tell you more about that work. And so the simulations and the modeling has been done mainly by my former PhD student, Lauren Melcher, who defended last year and is now a senior scientist at Pfizer, and then by Gregor Leach and Maya Nugent from Ray's lab and Michelle Chu from Mike's lab. And we just resubmitted the paper, and you can look at our most recent version on the archive. This is the preprint link, right? Okay, so why am I interested in this problem? So in my group at RIT, we are really interested in understanding the physics laws and concepts that underlie the robustness and the resilience and the ability to change of living systems and living materials, right? So we all know that living systems are the materials they're made up of are highly robust and resilient can change and adapt dynamically all the time, right? So for example, the first movie all of you have probably seen before, we have this white blood cell which is chasing this bacteria and pushing the red blood cell out of its way. It's really relentless in chasing the bacteria and can generate forces to move forward. It can also sort of change its shape as it needs to move through narrow constriction. It doesn't fall apart, and it can push things out of its way, right? So this cell is very soft and squishy, but at the same time, it's very robust and resilient. It doesn't fall apart, right? As it has to navigate all of this stuff, right? And then you can ask, what is sort of the physics that underlies the robustness and resilience of these kinds of materials? And then the second movie, you may or may not have seen before, but is my other favorite biophysics movie of an example of a living system that is very soft and squishy, but very robust and resilient at the same time, right? And all of these living materials are very different from current human engineered materials, right? Where you sort of start with a state and basically maybe you add motors or other activity generators, then your system goes to sort of a contracted state or some other state. But there is this, we don't see this kind of, resilience and robustness, which is evolving with time or which can switch back and forth with time, right? So if we wanted to make materials like this, in synthetic systems or in artificial systems, what would that look like? So the left-hand side is an example of what many of you would think it might look like. And my good friend and collaborator, Jenny Ross, this was certainly what she thought this should look like. But in fact, we also have examples of this kind of something, basically assembling, self-assembling, and then disassembling, and again assembling, etc. A good example of that in the living world. It's not just you see this, made by, basically in science fiction, but also the right-hand side example is an example of the ciliate. It's a single cell organism called Stentor ceruleus, and it's really very robust, again, to any kind of mechanical trauma, etc., right? And it can, again, if you make holes, it will sort of basically close those holes. If you chop it off, it will make new cells and regenerate and so on. So then if we want to ask is, can we build sort of synthetic biomimetic materials that can recapitulate this kind of robustness and resilience of living materials, but also basically that sort of, can go between, let's say, a self-assembled and a disassembled state, etc., right? And exhibit emergent physics, which we have not seen with current engineered materials, and materials in which we can sort of build in control and programmability, right? So what would that look like? Now, before we go to that, I wanted to talk about just the question, a little bit of robustness and resilience in soft, biomimetic soft materials. And the origin of that, right? So if you think about it, at a sort of mesoscopic scale, right? If we think about what are sort of the units of mechanical response for us, it's mainly cells and tissues, right? And cells and tissues, if you sort of looked at them if you zoomed in on them, they're all made up of multi-component or composite sort of soft material. So they have parts that are sort of more stiff, parts that are less stiff, parts that are sort of, will form networks or will form just bundles. There will be motors, for example, that can generate forces and so on. So there are lots of different types of things that come together to make these materials. And often the scaffolding in these materials is made up of very network-like structures, but these are also disordered networks. And I and my group and my collaborators, we have shown that for these kinds of systems, the mechanical properties, the mechanical resilience and robustness of these systems, we can use, the language of phase transition and what systems do close to phase transition to understand the properties of these systems. And the phase transition we have been sort of, we can sort of call upon here is the rigidity percolation phase transition, for example. And on top of this thing, of these materials being composite and disordered networks, they often also have inhomogeneous spatial structure, which is an additional knob that can sort of, help these materials to tune their properties even more. So just to give you some quick examples of what we have been doing here with these kinds of systems is we can sort of frame the structure, mechanical function relationships in these systems by modeling them as let's say, two interacting disordered networks that are sort of coupled to each other. And we call these biopolymer double networks. And we study sort of the rigidity of these kinds of networks. And what we have shown is that these biopolymer double networks basically bring, together different types of networks with different mechanical properties and in different concentrations to sort of basically give rise to composite materials that can be stiff or that can be soft and that can have different properties, such as, that can be sort of very ductile to fracturing, or that can be that will have brittle fracture, etc. Right? And so, but this is not the topic of my talk. I'm happy to talk to you about this later if you're interested. But just giving you an idea of the motivation of how sort of can we understand the origin of resilience and robustness in these systems. And we can also use these kinds of sort of, combination of using mechanical phase transitions in tandem with these kinds of networks and double network kind of structures to obtain insights into mechanics of soft tissues like cartilage and basically generate phase diagrams, which help us understand not just, what is happening, in cartilage tissue with, as you sort of degrade different parts of the tissue. Not only we can, explain the experimental data that we have access to, but we can also sort of explain what's happening in other parts of phase space, right? For which we don't have experimental data. We can make predictions. We can use similar types of models to also gain insights into the mechanics of networks of actin and myosin and microtubules and so on as well. Okay, but all of this is basically just saying that, yes, having this multi-component network-like structures can help make our systems, sort of be robust and resilient at the same time. But what about sort of changing their properties on a program schedule, right? Which is what I want to really talk to you about, and which is what I started out with telling you about, right? So again, we want to combine biopolymers from the cytoskeleton like actin or microtubules, right? But we want to combine them. We want to cross-link them now using cross-linkers that basically involve these circadian rhythm proteins or clock proteins from cyanobacteria. And the goal is to make materials that can basically go sort of repeatedly and sort of in a scheduled way between cross-linked and uncross-linked states, or between softer and stiffer states. So to tell you a little bit about the cyanobacteria clock proteins, by the way, we all have clock proteins, right? Which is why our metabolism can sort of proceed at different rates, depending on where we are in that clock, right? So we all have biological clocks. So cyanobacteria have clock proteins called Chi-A, Chi-B, and Chi-C, which come together and form a complex like this, right? At the sort of peak of this 24-hour cycle. And at the trough of the cycle, what happens is this Chi-A, Chi-B, Chi-C, they will go apart, and now they are not connected together anymore, right? And what we wanted to do is, our goal was, we are going to coat some fraction of our clock proteins. When they're in this state, we will make them a cross-linker by sort of coating them with something that will make them sticky to the cytoskeletal polymers, right? So this is sort of the goal. But we soon learned that this is actually a very complicated system. So we wanted to then do something which is proof of concept. And instead of doing this study with biopolymers and these cyanobacteria clock proteins, we decided we will do them with colloids and cyanobacteria clock proteins first. So that's what then we set out to do. So now, once again, our circadian clock toolbox consists of these three parts of the cyanobacteria clock protein complex, right? So we have Chi-A, we have Chi-A, we have Chi-C, which forms this hexamer, and we have this Chi-B. And then what we do is we biotinylate, we coat some of the Chi-Bs with biotin, streptavidin. We also coat the colloids with biotin. And what this means is that on a 24-hour cycle, when these clock proteins have formed this kind of a complex, and then we have these Chi-Bs on this, which have sort of this biotin coating on them, they can basically then go and stick to the colloids, which are also biotin coated, and these guys can then act as cross-linkers between these colloids, right? And then on the other side of the 24-hour cycle, when these clock proteins have all sort of disassembled and they are not in a complex form anymore, then the colloids are not connected by this cross-linker anymore, and then now everything is sort of unlinked, right? So this is sort of, this is what the toolbox looks like. And if you, in the fully connected state, this is what our colloidal network would look like. And in the sort of disassembled or mostly disassembled state, this is what the system looks like. So before we add any colloids to the Chi-ABC toolbox, we wanted to first see, if you biotinylate your Chi-Bs, how does that affect this cyclic going between forming a complex and not being able to form a complex abilities of Chi-ABC, right? So what we find is that if we do not coat any of the Chi-Bs with the biotin, of course, this is basically just the wild type, Chi-ABC, then we see oscillations like we would see anyways, right? If we coat 55% of them with biotin, then we still see oscillations, but the oscillations, the amplitude of oscillations will attenuate, right? So you'll have a Chi-ABC complex when you're at the peak, and when you'll have like Chi-A, Chi-B, Chi-C will be separated out when you're at the trough, etc. And what we see is that when we coat 80% of our Chi-Bs with biotin, then they mostly stay in this complex form. They don't really separate out in the complex, right? And now all of this is without adding the colloids. So next we actually add the colloids, and what do we see in our experiments? So what we found is that if you don't have any biotin-coated Chi-Bs, so then we will have no cross-linkers or no bonds. So the colloids will not form any clusters. They will just stay separate, right? And when you have 80% of the Chi-Bs are biotin-coated, then we will have this, they're mostly in the complex state, right? And then they will act as permanent cross-linkers, right? And you'll get really large colloidal clusters. But now in the intermediate state where, a certain, like 55 or so percent of the Chi-Bs are biotin coated, what we found in the experiments is that we don't really see oscillations. We still see that the size of the colloidal clusters progressively keeps growing. So we wanted to understand what is really going on here and what is the mechanism, right? And to do that, we turn to simulations. So the simulations that I'm going to describe next are looking at 3 different cases. One case when there are no bonds. So this is basically the wild type situation where the Chi-Bs are not coated with biotin at all. Then when we have permanent bonds where, a large fraction of the Chi-Bs are coated with biotin. And the time scale here is governed by the clock protein, complex formation time scale. And then we have the situation where you can have both attachment and detachment that are governed by, again, clock protein, complex formation, and disassembly timescales, right? And with this sort of simulation model, then we say that anytime 2 colloids are within a certain distance of each other, which is equal to or less than the distance, which is basically their diameter plus the Chi-ABC complex length, then a bond will form between them with a certain probability, and this probability is going to depend on, again, it's governed by the timescales of Chi-ABC complex formation. So with that, we first looked at the 2 sort of idealized situations, right? When there is permanent bonds and when there is no bond. So when there is permanent bond, once again, we saw in our simulations that, we see clusters. So before I tell you about clusters, let me tell you what this color bar means. So in the color bar, when you are on the dark side here, it's basically you're looking at timescales of about 1 hour from the start of the simulation or start of the experiment. And when you're at this light cyan and then almost white, you're looking at what's happening at the 28th hour mark, both in our simulations as well as in experiments. Same thing for this case, when you are at 1 hour mark, it's really black. And at the 28-hour mark, it's like getting yellow, 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 and close to white, right? And what we see is that, at smaller times, you can see that there are very small size clusters, both in experiments as well as in simulations. And this is the permanent bond case. But as we go to larger and larger times, we have really large clusters of these colloids now, right? And these are growing with time. And in the no bond case, again, no bonds form because, the Chi-ABC complexes cannot act as crosslinkers because they are not biotin-related. So we don't really see any cluster formation either in the simulations or in the experiments. Now, one of the things you would notice is that in the simulations, the cluster sizes seem bigger than in the experiments. And we think that's happening because the simulations are in 2 dimensions, while the experiments are in 3D. So in experiments, the colloidal clusters can also sort of grow in the 3rd dimension. And in the experiments, in the simulations, they're only, sort of growing or forming clusters in 2 dimensions. So we sort of have a larger size in 2D, or rather than when we are looking at these, 2D versions of 2D slices of 3D images. So now, once we had these, we then wanted to look at things a little bit more quantitatively. And to do that, we looked at the spatial autocorrelation function following the same method in experiments and in simulations. And I'm here showing you the spatial autocorrelation. So this is not the radial distribution function for particles, but we are looking at autocorrelation functions between pixels with given intensities, right? And what we find is that, once again, in the no bond case, this, there is really no correlation. It dies down pretty fast. But in the permanent bond case, as you go to larger and larger times, of course, we get larger and larger clusters. We see more and more correlations. Now, if we calculate the correlation length from these graphs, then we can see in the no bond case, the correlation length is 0 or close to 0. But in the permanent bond case, the correlation length is going to grow like this, right? And this is all from simulations. This is what I'm showing you here. We can also look at the colloid connectivity number. And that's basically the number of nearest neighbors colloid has, right? And again, in the permanent bond case, this will be very close to 6 because we are in 2D, and it indeed asymptotically approaches 6. And in the no bond case, this is 0. And we can see that, if we calculate a quantity, which is the fractional clustering index, which is basically the values like the correlation, length, etc., are normalized by their maximum possible value, we see that there is very good agreement for this permanent bond case between the simulation data and different measures that we can get from experiment, right? Including the correlation length from experiments.
Mo, about 4 or so more minutes, just to let you know.
Okay. All right. So we have 4 minutes left, and I should switch to the more interesting part of the story. So first of all, what is really going on in the case, right? Why did we not see oscillations when we had 55% of our Chi-Bs sort of biotin coated? And what we think is happening is if a pair of colloids were only connected by one Chi-ABC complex like this, then maybe we would have seen some oscillations. This is what our simulations are suggesting. But the colloids will never form large enough clusters, right? Because they start, clusters start dissolving before they even have a chance to form. However, if the valency is not 1 bonds, but 2 or 3, then basically we will see colloidal clusters forming, but these clusters will now grow in size, and they will actually not dissolve, right? So that's what we find. And the time scale of this self-assembly is governed by the time scale of Chi-ABC complex formation. And now it's also suggesting bonding valency, right? So the connection between the colloids is not monovalent. It's larger than monovalent. And we also saw that this sort of explanation it basically is borne out if we look at, again, the simulated correlation lengths and the experimental correlation lengths and so on. Okay, fine. So we understand why we are not seeing oscillations. But is there a way to see oscillations, right? So under what conditions might we see oscillations? And to predict that, we first turned to our simulations. And what we found that instead of having the valency, right, or the number of Chi-ABC crosslinkers connecting a pair of colloids be 1 or 2 or 3, if it's something between 1 and 2, so let's say 4/3 or 5/3, then we can see that this colloidal connectivity number, right, show actually an oscillation, which suggests that in experiments, if we sort of replicated that, we might see the system going between this cross-linked, the state with large clusters and state with no clusters, right? So then we indeed and before we did that and tried to do that in experiments, we were like, okay, let's also actually calculate the correlation length and the fractional clustering index in our simulations. And we find that, yes, our fractional clustering index does show these oscillations. And of course, it also shows up in our the configurations of the simulated clusters, right? So then we actually went and did this in experiments. And so in the experiments, first we checked that the Chi-ABC complex formation is not impacted, if we try to sort of basically get our colloidal, basically if we try to have colloids present with Chi-ABC with the kinds of concentration which will mimic this, right? And we found that, yes, we still see oscillations. So this is good news. So what did we do in the experiment? So what we found is that if we have concentrations of Chi-Cs, which is 100% or 50% that of the rest of Chi-As and Chi-Bs, so we will get complex a lot of the time, then we will see this kind of large cluster formation and things will never dissolve. However, if we get our concentration of Chi-Cs to 25% times that of the other guys, so which means that we will not have complex formation as often, and that will mimic these kinds of valencies that we were predicting with simulations, then we will indeed see these kinds of material oscillations where we go between not having a colloidal cluster to a connected colloidal cluster to not having a colloidal cluster to a connected colloidal cluster, right? So with that, I will come to the summary and outlook for my talk, for this talk. And hopefully I have given you a glimpse of how systems, biomimetic and biological materials being composite and multi-component sort of facilitate robustness and resilience and tunability in these materials. But more importantly, hopefully, I have also given you basically shown you how we can use biological timers or biological circuits to control the self-assembly and disassembly of synthetic materials in a sort of persistent way. And we think that this might help us get insights into how to make autonomous materials with control and regulation, right? And so with that, I'm going to thank my group and my collaborators and all of my funding sources. And again, the names that are in blue are names of folks whose work I have shown here in my talk today, and the names in red are the names of my former PhD student and my collaborators who are directly involved in the main part of the talk. Thank you, and I'm happy to take any questions.
Let's thank the speaker for a wonderful talk. And we have time, I guess, maybe for one question, and then we can save more for after. Feel free, Sriram. Feel free to.
Yeah, Moumita, I just trying to, do you have any idea how you can make the oscillations sort of more cleanly timed? I mean, that is more sharply periodic in the experiment. Do you have some insight from the simulations?
So the thing is, yeah, I'm going to stop sharing and maybe I can see. Yes. So I think what we are so the thing is right now, the colloids or the crosslinkers we are using, they are on a 24-hour cycle, right? And the way to make this more robust, I mean, right now we have only tried concentrations, which mimic the 5/3 and the 4/3. But these are sort of, just things that we saw work. But we can actually try to optimize this and see what valency actually gets us both, large residence times in these two states, as well as large differences, right? Large amplitude as well as large residence times. So we can get that from simulation, and then we can try to get the right concentration of chi-Cs versus chi-As and chi-Bs, which will give us that, that kind of valency, right? So that's one thing. The other thing is that changing the time period itself, like right now, so you can make mutants, or you can make mutants of these clock proteins, which can have, time periods that are somewhat different from what we have right now. So hopefully that answers, at least starts to answer some of your questions.
Sure, I'll catch you. I'll ask you more later.
Mm-hmm.
Thanks.
Thank you.